Hello, my name is Gavin Gordon. I'm the Vice President of Clinical Market Development here at Akoya Biosciences. I'd like to thank the conference organizers for the opportunity to speak today, and I'll be talking to you about how spatial biology approaches are being used to drive innovation in predictive biomarker discovery and validation in oncology, specifically immuno-oncology. Precision medicine approach is largely developed out using NGS and targeted therapies is moving to immuno-oncology. The reason is because progress here is going to require a spatial biology approach to biomarker discovery, not just to characterize all the phenotypes and the complexity of the immune repertoire, but also understand where these cells are in the body. Are they in the tumor? Where in the tumor microenvironment are they? What other cell types are they interacting with? All of these are important questions in determining how a tumor is going to respond to any given immuno-oncology drug. It's not possible to get this level of detail and complexity of analysis using a traditional NGS approach. Clearly, a protein approach that incorporates a spatial component is going to be needed. Multiplexed immunofluorescence is one such approach. So what evidence is there, if any, that this type of approach might be useful in immuno-oncology biomarker strategies? Well, in this JAMA Oncology paper, representing a meta-analysis of almost 60 studies, the authors set out to answer that exact question. Looking across all of the papers, encompassing almost 10,000 total patients, 10 solid tumors, they compared multiplexed immunofluorescence to tumor mutational burden, gene expression signatures, as well as conventional single marker PD-L1 IHC to identify which of these platforms had the best predictive capability for overall survival for immuno-oncology drugs. The authors found, encouragingly, that multiplexed immunofluorescence had as a single modality the best predictive performance, as shown via these ROC curves in the lower left-hand side of the screen, where MIF is represented by the blue curve, protein spatial phenotyping. And of course, the further these curves are shifted to the left, the better the test. So this was a really interesting study. It certainly wasn't conclusive by any means, but it did provide evidence that this type of approach could be useful in immuno-oncology. Akoya has two primary multiplexed immunofluorescence platforms that are being used across the medical and scientific community, including by our next speaker, to do exactly this. Look at protein spatial phenotyping in oncology. The Codex platform provides the ability to look at almost 50 markers simultaneously, but at relatively low throughput, a few samples a week. On the other end of the spectrum, Fanoptics, including the high-end Spectra Polaris, allows interrogation of up to 9 protein markers, but in tens of samples each week, hundreds of samples each month. So there's clearly some trade-offs here between the number of markers that you can look at and the number of samples you can evaluate them in. So drilling down in Fanoptics just for a moment, because this is the platform that provides most of the clinical and translational researchers with their data, you can see the trade-offs shown here. And what most folks are finding is that 6 to 7 markers is really the sweet spot for translational studies. It allows a whole slide scanning in about 10 to 15 minutes and analysis, and allows the evaluation of hundreds of samples each week. So it really forms a nice way to test hypotheses that are identified using the Codex and other platforms in discovery-driven research. So what sort of issues does a spatial biology approach to biomarker discovery encompass? Well, let's start with the goal. The goal is really a test that provides an operator-independent algorithm that encompasses the data in millions of cell types and an almost infinite array of potential spatial measurements and drills that down to a single actionable and optimum score. A plus or a minus, a yes or a no. Something that provides a readout to enable assignment of therapy in immuno-oncology. There's at least 3 scales you can explore a spatial approach, from top to bottom, or low resolution to high resolution. You can look at the tissue architecture the tumor, the stroma, other non-tumor elements in the tissue section. You can look at the tumor microenvironment, different areas of tumor that have different combinations of immune cell infiltrate and tumor cells. And then, of course, you can look at single cells and how they interact and engage with other single cells. So to give you a sense of the problem or the complexity that this approach entails, here's just a thought experiment showing as many parameters as I could think of that when multiplied together give you billions of possible parameters to evaluate for an optimum signature. So this is clearly a daunting task, which is to say that it's incredibly complex. However, as our next speaker will show, and other investigators have shown, is that simply looking at a relatively small number of these parameters and implementing it in a clinical and translational research setting has the ability and potential to improve dramatically on what you can obtain using traditional biomarker strategies in immuno-oncology. One example is a study out of Johns Hopkins, their AstroPath initiative, where vector-polaris spatial phenotyping data was used in a study in melanoma to look for spatial signatures. They used the tools at Johns Hopkins that were traditionally used to study the night sky and turned them inward into the tumor microenvironment. This was effectively a big data engine, producing a lot of data that was ultimately validated in melanoma, and a signature was found in a relatively rare phenotype, an infrequently studied cell type, FOXP3, PD1, and CD8 marker positive, I believe. Where these cells were located in the tumor and how many of them there were was predictive, more so than IHC, TMB, or MSI. And all of this algorithm development could be implemented in a vector-polaris and standard Inform software. And so at this point, I'd like to stop the introduction and introduce our next speaker, Dr. Sebastian Morowitz from the Leibniz Lung Center in Germany, who'll be exploring how some spatial profiling work was done with he and colleagues in lung diseases using a vector-polaris and multiplexed IHC and FFPE patient samples. Thank you very much.
Hello and welcome everybody to my talk. First of all, I would like to thank Gavin for the introduction and the organizers, as well as Akoya Biosciences, for having me here with you. I would like today to talk about a showcase for hypothesis-driven analysis on cohort level using formalin-fixed paraffin-embedded patient samples in the context of lung diseases. So, disclosure at first, I have no conflict of interest to disclose, and all images were acquired on Akoya Vector Polaris using motif scan at 20x magnification. Image analysis was conducted using Akoya Inform software and Phenoptics reports R package, as well as a Tidyverse GTPR for data presentation. As an alternative to Akoya's Inform software, I could also alternatively highlight QPath open source software developed by Pete Bankhead, currently in Edinburgh. Please browse this webpage to get a hint on the software, or read the paper published in Scientific Reports 2017. It is an awesome piece of open source digital pathology software. So today's talk will be in a Lowplex setting. So speaking of Lowplex, multiplexed immunochemistry, multiplexed immunofluorescence, we will see pictures of images with panels less than 10 or less than 20 markers. And this might sound currently a little bit outdated, but I would like to show you the benefits of using Lowplex, but in a high or larger cohort volume. So speaking of Lowplex, we have CD20 for staining of B cells. We have CD8 for effector T cells. There is CD3 as a pan T cell lineage marker, CD68 for alveolar macrophages, FOXP3 for regulatory T cells, and for instance, pancytokeratin for detection of tumor cells as well as epithelium. Although, or by using a Lowplex setting, it has some certain advantages. So the combination of few markers that co-localize, for instance, CD3, CD8, or FOXP3 on the same cell or same compartment, creates already an abundance of phenotypic combinations. So these combinations are further amplified by the amount of tissue segmentation classes. So how many different morphologically distinct categories are detected and defined by the user, these categories will further amplify the already observed phenotypic combinations. Furthermore, those spoken phenotypic combinations with respect to different tissue segmentation results are also amplified by spatial metrics. So for instance, the mean distance of a CD3 positive, CD8 positive cell to CD20 positive B cell can be computed in tumor and stroma compartment as well as any other detected tissue segmentation result. So this can lead to dozens, if not hundreds of variables without any clinical data or metadata. So once it is merged and annotated with clinical and patient-centric data, feature selection is a crucial step. But nevertheless, the whole workflow using the Vector Polaris system with the downstream software is a workflow that's manageable for non-data scientists without any coding knowledge. So it is suitable for the average wet lab Joe. There are different ways to analyze spatial data in the context of paraffin-embedded tissues. I want to speak today about 2 or 3 of them. The list or the ways to analyze spatial data that is very likely not a finite list, but just a short glimpse of what information can be deduced out of the tissue. So as one of the very first steps in image analysis, there's usually a kind of tissue segmentation where the user trains an algorithm to differentiate the different morphological compartments of a tissue. For instance, the stroma can be differentiated from the tumor islands, or blood vessels can be detected, or necrosis, or anything else. And by having a tissue segmentation, it will create a border, and that border can actually be used and applied to compute cellular distances. For instance, the distance from a border, for instance, for the distance of a CD8 from the tumor margin that penetrates into the tumor tissue. So that distance can be calculated to get information on how deep an immune cell was able to invade into the tumor cell or into the tumor compartment. Furthermore, the next way to analyze spatial data would be to focus on a certain cell, a certain cell type, a cell of origin, and to look then which other cell types, as indicated by different histological markers, are at a given distance related to that cell. So by that, we are able to compute cellular distances in between cells. For instance, the mean distance from a CD8 cell to a CD68 cell. Furthermore, we are able to start again from a cell of origin, a cell of interest, and define a radius to create a hypothetical circle around that cell of origin, a cell of interest. And within that given circle, we can compute the average amount of cells being within that radius. That actually leads us to the measurement of cells within a given radius. For instance, the mean number of CD20 positive B cells around an CD3 cell. Today, I would like to speak about 2 of these spatial metrics. The one would be the cellular distances in between cells, and the other one would be cells within a given radius. And I would like to show you in about 2 Lowplex settings, what kind of data can be analyzed by that and what kind of questions can be asked and what results will be presented. So speaking about cellular distances in between cells, there are multiple ways to show or plot that kind of data. One of that actually is a density plot, as people might be already familiar with, in form of histograms from flow cytometry data. So on the x-axis, you will usually find a mean distance to a cell type of interest. And on the y-axis, there usually is a scale that's called a density. And that scale is related to abundance of cells. And within that given plot, we can actually define a cell type 1, a cell type 2, a cell type 3, and depending on the height or the height of each of those peaks is being defined as the amount of observations where a certain cell type, for instance cell type 1, is at a given distance to the cell type of interest. And this is then computed for hundreds of observations among different patients within a cohort. And because we have multiple observations, we can calculate the mean distance within a given cohort for a cell type 1 to the cell type of interest. And we can calculate furthermore this for any other cell types. And the combinations of these mean distances of one cell type to a cell type of interest can be somehow used as a so-called spatial fingerprint. And the spatial fingerprint doesn't necessarily have to be the same within a cohort of patients. So these spatial fingerprints might be very different in between patients, and that information of mean distances to cells of interest can be used to probably draw clinical or maybe therapeutical conclusions. So spatial metrics as a numerical unbiased representation of morphological processes are somewhere like turning an image, what we have in mind by looking at it, into a number that can be further processed and doing downstream analysis in an unbiased and reproducible manner. So speaking about nearest neighbors and distance analysis, there are certain caveats to consider. For instance, take the mean distance of CD20 positive cells to CD68 positive cells. And then you plot the mean distance of CD68 positive cells to CD20 positive cells, and you could see that they correlate very poorly. There's no real linear correlation among these. And so distances are not necessarily reciprocal. So it might really depend on the origin of a cell or from which cell to another cell a distance is calculated. Coming back to the analysis of lung cancer or non-small cell lung cancer tissues, on the next few slides, I would like to show some data that has been mainly deduced from the stromal part, because I was interested in immune cells and distances in between immune cells. So when I applied that Lowplex panel to a couple of cohorts of non-small cell lung cancer patients and calculated the mean distances in stromal compartments, it was clear in the end that the patients organized themselves in an unsupervised manner, and that resulted in 3 different groups of patients who behave differently. And these 3 groups are probably driven by 3 clusters of cellular distances that I believe are the driver behind that variance and the driver behind how these patients behave differently within the stromal immune cell composition and immune cell organization. So one of the three clusters would be the mean distance to CD20 positive cells. The next cluster would be the mean distance to FOXP3 positive cells. And the third cluster would be the mean distances of cells towards CD68 positive cells. So by aligning multiple variables from the distance calculations, one can also observe distances or cellular events that behave very comparably. So here we have, for instance, CD68 positive cells, or the mean distance of CD68 positive cells to CD20 positive cells. And we have on the x-axis CD3 positive, CD8 negative, which are likely to be CD4 positive, and their distance to CD20 positive cells. And again, here you can see that both distances appear to be very, very comparable in stroma compartments as well as tumor compartments. So CD68 positive cells probably have a comparable distance to CD20 positive cells as CD4 positive, CD8 negative T cells, which is of interest, I would say. So focusing again on that cluster of cells having a mean distance to CD20 positive cells, and that drives or is probably a driver of that group accumulation among non-small cell lung cancer patients, we can have a closer look on what's going on with the mean distances to CD20 positive cells. So here we have plotted for the stroma and the tumor from patient material from the tumor center and the tumor margin. We see how the patterns actually look different in stroma and tumor. And I don't want to go into too much detail, but if we just focus on CD20 positive cells and their distance to other CD20 positive B cells, we can easily see that for most of patients here, there is a very short distance in between B cells. So here we have the 100 micrometer distance from a B cell to another B cell, and the mean across the cohort is somewhere well below 50 microns. So apparently B cells tend to stick to other B cells very frequently among patients in the stroma compartment, but not in tumor compartment, as you can see here, where the density is way more evenly or way more differently distributed. So we then calculated the mean distance between CD20 positive to CD20 positive cells, and it's no surprise to see that there were statistically significant differences in between stroma and tumor in one cohort, and with a similar trend in an independent different cohort. Furthermore, if we go back to the images and how they actually looked like, you can see here for one patient having a very short distance in between CD20 positive to other CD20 positive cells, that the cell classifier will highlight those stroma organizations, and it can actually then use the calculated mean distance between a B cell and another B cell and use that for survival analysis. And you can see here for overall survival or disease-free survival in a cohort of surgically treated non-small cell lung cancer patients, that patients who have a short distance in between B cells have a longer overall survival and a longer disease-free survival. So the short distance of CD20 to CD20 cells is likely to be represented by tertiary lymphoid structures, and these structures have been well described to be an independent prognostic marker for patient survival and even immune checkpoint therapy success in different types of cancer. The beauty of this actually is it is independent of any image annotation, any subjective scoring or rating. It is just based on the calculation of cellular distances, and that actually brings back the beauty of an image connected to the real world numbers. So coming to the next spatial metrics, cells within a given radius, I would like to show some very short results of a study we published recently. So speaking about cells within a given radius, we applied in the laboratory of Bernie Fox in Portland, Oregon, 2 panels to a cohort of non-small cell lung cancer patients, where we first of all looked into T cell proliferation and expression of PD-L1 on macrophages, FOXP3, Tregs, or CD8 cells, all of them combined. Also with phosphorylated SMAD3 as an activation marker for activated transforming growth factor beta signaling. We use punches from different regions of patient tumors and applied a digital pathology workflow to it, where we use machine learning based feature selection for variables impacting survival outcome, and applied cells within a given radius of other cell types. And that actually, based on findings of the Fox lab, where they found out earlier in 2017 that in oral squamous cell cancer, a radius of 30 micron around CD8 cell, and when there are high numbers of PD-1 expressing cells in tumor or stroma within that 30 microns around CD8 cells, patients tend to have a shorter survival. So we applied that to our cohort on different observations, where we actually focused more on the effect of FOXP3 regulatory cells within a given radius around CD8 cells within the tumor, so not in the stromal part. So we wanted to see what kind of signals FOXP3 positive cells are likely to exchange with the effector T cells. And we also used accessory markers like PD-L1, phospho-SMAD3, and combinations of those to compute cells being around CD8 positive cells that have either one of the markers or both of the markers. And you can see here that a high number of FOXP3 cells, independent of any other accessory marker, is already a good indicator for a shorter disease-free survival or a shorter overall survival in non-small cell lung cancer patients. But once you take PD-L1 into consideration, the group or the cohort stratifies even better. And combined with SMAD3, or with phospho-SMAD3 and PD-L1, the patients having a high number of PD-L1 expressing phospho-SMAD3 positive FOXP3 cells around CD8 cells is already a very good indicator for a very short survival and a very quick relapse. But once you take only phospho-SMAD3 into consideration and FOXP3 regulatory T cells, you can stratify the cohort to patients who have a very, very early relapse. So I would like to sum up the previous slides with, or I hope I could convince you that the use of spatial profiling in Lowplex settings enables a hypothesis-driven analysis on cohort level for biomarker discovery, for instance, as the cellular distance, and also spatial profiling aiming at activation markers or ligand-receptor interaction might be a promising tool for development of therapeutic inhibitors. So there's been recent development, for instance, in dual targeting of TGF-beta PD-L1, and I'm very convinced that multiplex imaging, multiplex immunohistochemistry, and digital pathology workflows actually really might drive those efforts. So I would like to thank the people involved in these studies that are our clinical partners at different academic clinical centers. We have close collaboration with Raj Kumar Savai and Max Planck Bad Nauheim regarding TGF-beta targeting. We investigate also further phospho-isoform signaling with a Ph.D. student in our lab. And I'm very grateful to have spent some wonderful time in Bernie Fox's lab in Portland, Oregon, where we discuss and work a lot on multiplexed immunohistochemistry applications with regards to immune checkpoint blockade. And we also had some great time with Patrick Mickinker in Estrel in Uppsala, where we focus mainly on cancer-associated fibroblasts and non-small cell lung cancer tumor microenvironment. So thank you very much for your time, and I'm happy to take questions during the later on Q&A session.